Hello, students, and welcome to my screencast on cell differentiation and meiosis. This will wrap it up for this unit. It's Chapter 10, Section 4 in your book, and Chapter 11, Section 4. Chapter 11, Sections 1, 2, and 3 are genetics, and I kind of like to do meiosis and then lead it into genetics. The products of meiosis are what's going to go on the top and sides of those Punnett squares. All right, I'm going to use some images from Khan Academy, and once again, this is not for commercial purposes. So here are my kids wearing these cute banana shirts. Sorry, I couldn't find Rory's banana shirt picture, but he has one. Thank you, Auntie Sarah. So they're all, why are they different? Why is my son different than my daughter, who is different than the other son, etc.? And it's due to meiosis. That's the overarching phenomenon. So here we go. Why also do we have specialized cells, right? You have eye cells, you have cells for smelling that have olfactory senses, you have specialized cells for taste in your mouth, you have your liver cells for processing items, you have your heart cells that will keep your heart going and pump blood throughout your body. So that kind of answers it, right? The reason we have specialized cells is it gives us the emergent properties of being human, right? It gives us the ability to go off and do things that a bacteria or an amoeba can't. Amoeba can't get up, walk around, and go run, taste some track, or play taste some soccer or basketball, but you can. All right, so we all start out as one fertilized cell called a zygote. That cell can then, those cells differentiate into these types. And notice how different they are. Remember, the structure of a cell is going to lead to its function. The structure of these intestinal cells let it absorb lots of nutrients. So it goes from your intestine and it can absorb them and then kick them into the bloodstream to send it everywhere. For example, glucose. All right. What causes them to differentiate? This is tough. This was a long time ago. We went over this way before DO. So see if you remember. Hopefully you got that. One of them is asymmetric distribution of transcription factors. What that means is that it's not even in the initial zygote. The proteins that are in the zygote are not evenly distributed. So when it divides in half, one part of the zygote is going to get more of a certain type of protein than the other. This would be an example if, if they were all evenly distributed, because I believe this has 3, 6, they each have 8, right? But this is what happens for real inside of us. You're going to have different ones in different cells. It's then going to code to make that cell different. We can also, once that starts happening, right, nearby cells can send signals. You're familiar with this, ligands, which can then cause a signal transduction pathway and access DNA to cause the cell to be like this one. So a cell receptor or a signal can cause a cell to become a certain way, or even the environment. So this is pretty cool. Depending upon the environment, it'll affect Himalayan rabbits whether or not certain parts are black or white. So one thing that confuses students, right? Every cell in your body has the same DNA. They just access different genes to lead to their different structures. Here, this neuron is accessing genes for being a neuron, whether this liver cell is accessing genes that help it to break down poisons. So the cells in my eye are activating different genes in the cells in my mouth, but they both have the same DNA. The only place that isn't true are some of your immune cells because they're special, and also in your reproductive sex cells. Here's an example of an adult stem cell. So it can become all these different types of blood cells, right, or cells involved in your blood. And so adult stem cells can do that, but they can't become anything. They're kind of restricted. They're not as versatile as embryonic stem cells. Now, we have had some success in getting cells to go backwards, and it's called induced pluripotent stem cells. But there's still great debate, and there's great debate on whether or not we should do embryonic stem cell research, and I think you should research that outside of this video. Who knows? Maybe it'll come up at some point. Okay, let's jump into meiosis. So we talked quickly about how cells become different. Now we need to talk about how we make our different sex cells, the different sperm and eggs. Let's pretend that this is mom and this is dad. So red is mom, purple is dad. These are homologous pairs of chromosomes. They're the same size and they have the same genes on it. Gene A, gene B, and gene C. But notice, students, that they have different versions of the genes or different alleles. Mom here has big A, and dad has little a. So they have different forms of it. Mom, big B, dad, little b, and I believe this is big C, little c. It's a little hard to read for the C's. Here, they're going to engage in crossing over. So they're swapping that allele or that form of the gene. So now these chromatids are no longer identical. This is one of the unique genetic variety products of meiosis, which is very important. So here's a picture of it. I'm going to go a little fast through this because I just did this in the homologous chromosome video, and it's the exact same image. This is 2n is equal to 4. So if we're doing meiosis, we want to make n is equal to 2. We want to cut that in half, right? So that we can keep doing the life cycle. Then it's going to find an n is equal to 2. They'll come together, and that'll be 2n is equal to 4. So here's my homologous pair. Here's another homologous pair. They're going to line up. This will be completely random. This is another point of genetic variation. This is called the independent assortment, or it's also called the random assortment of homologs. So they've crossed over. They line up in the middle. They get pulled apart. Notice how the homologous pair gets pulled apart. The homologs, or one from the pair, gets pulled to the opposite end of the cell. The sister chromatids, however, stay together. In your new cell, notice how many chromosomes there are. 1, 2, 1, 2. So we've already cut the chromosome number in half. We've become haploid by the end of meiosis 1. Now it's just a matter of getting it so that we have just one chromatid of each chromosome in the cell. We wouldn't want 2 chromatids in the sex cell because then we would have too much DNA, right? We only need 1 chromatid. So here they are. There's 2 chromosomes here. We line them up in the middle, just like in mitosis. We pull them apart, and now you get your 2 chromatids in each cell. Now when they combine with another cell, another sex cell, then you're going to get the other chromosome number 1 and the other chromosome number 2. Then they'll be able to replicate, and you'll have the baby, you'll have the zygote, and then they'll be able to undergo mitosis. So let's see, big picture. 2n is equal to 4. 4 chromosomes, 2 in each of the resulting cells. And you only want 1 chromatid in each. All right, here is a model of it with Playdoh. So I encourage you to try and make something like this. This would be the homologous pair, same size, same genes, dad, mom. Okay, they copy each other they get replicated during S phase. Here you go. Now they're going to engage in some crossing over. They've crossed over. They've exchanged some alleles. They separate by the homologs move to each side. So now we have n is equal to 1. We started with 2n is equal to 2. Now we have n is equal to 1. They're going to line up like mitosis and divide again. You have n is equal to 1. Right? We only needed 1 chromatid from there. If we had 2 chromatids and then it underwent mitosis, you would end up having 4 for that 1 chromosome. So maybe just draw it out and you can see why it would get crazy. So here we just need 1 chromatid. It's going to uncoil, and we have the DNA we need. So you, in your cells, in your sex cells, you have n is equal to 23. Now, we did go over non-disjunction in class, and unfortunately, I don't have pictures of it in this video, so you're going to need to research that. But there can be errors during this process in meiosis 1 or 2, and that can lead to an improper number of chromosomes in these sex cells. All right, this is from the Amoeba Sisters. I highly recommend you check out their chromosome counting video if you're confused about what's going on here. Notice how meiosis is going to make all different ones and mitosis makes identical. All right, students, study hard for that test.